Well, not hearing anything, then I guess we will get started. So my name is Kevin Smith, and I have the pleasure of introducing a group of folks that are going to talk to you about some of the work that we've been doing with Silphium or Silflower. So you'll hear us call Silphium or Silflower either of those two names. Silflower is sort of the common name that many of us in the group are starting to use more often, but we have no legal trademark status like Kernza. So you'll hear us use the word both Silphium and Silflower. So I'll give a little bit of very brief background, and then I'm going to turn it over to the rest of the folks who actually do all of the work. So let's go to the next slide. All right, so just for those of you who are not familiar with Silphium, it is a member of the Asteraceae. It's also a component of prairies in the central part of the U.S. So it's native. It's a perennial. You can see some pictures of it here, and it is a crop that has been under domestication for the past 20 years or so. So in terms of domestication age, it is in its infancy. But it was identified as a promising species for domestication as a crop in agriculture for a number of reasons. It's perennial, so it fits under the framework of the Forever Green Initiative. It has a very deep root system and was identified very early as a very drought-resistant plant. It has a shape and structure that makes it amenable to agriculture, upright with flowers and seed bearing towards the top of the plant. Nice, reasonably good seed size for a native plant, and oil content and composition that is similar to sunflower. So in terms of a crop, you can think of it as a sort of perennial version of sunflower, and also very pollinator friendly. So we think it has a number of interesting opportunities. Next slide. However, it has some challenges as well, obviously. Otherwise, we wouldn't be part of this initiative because we all have challenges, right? So it has a long breeding cycle time. So if you plant Silphium by seed in the spring, it will grow in a rosette form close to the ground for the first year, over winter, and then it doesn't actually bolt and produce flowers and seeds until the second year. So from a breeder's perspective, especially a barley breeder who's used to planting seed and getting seed back in a couple of months, that's a long time. Harvestability is a challenge. It can shatter. The flowers develop over a long period of time. So heterogeneity of flowering is also an issue with harvestability. The plants tend to be fairly tall. So in breeding, we're thinking about, things like plant height and maturity. It's an obligate, it's an outcrosser. So again, unlike barley or wheat, we can't just self-pollinate and produce big bags of inbred homogeneous seed. So we would like to have a way to propagate it or clonally propagate it for various reasons. It has been identified to have some disease and pest issues, particularly in Kansas, where a lot of the work really initiated, and where there have been plots established for long periods of time down there. There seems to be less issues with disease and pest as you travel further away from Kansas, but that's something that we're interested in and we recognize as a potential challenge. And then of course, we're always trying to improve yield. And as a perennial, we want to maintain that yield over a period of time. So those are just some of the challenges. So this is a little pictogram that Kelsey put together from our group. And it follows the sort of hallmark of the Forever Green Initiative crops, in that we don't think about the research that we do in our own little silos. We think across disciplines, but not only across disciplines, we think across supply chain. So we think about the whole spectrum of research activity from basic research to end user. And this is just our little representation of that. We're going to focus today on the things that are on the left side of that circle, but that is not to say that we don't think about the other parts of this research circle, including outreach activities, ecosystem services, and supply chain issues. We certainly think and talk about those things, but our research is really focused on the things on the left to get us up and running to the point where we can still really start to think about those other things. So you see, so what we're going to do today is have a series of lightning talks, essentially from the 4 graduate students and a postdoc. So Sienna, John Hill, Yuki, and Kelsey are the graduate students on the project, and Andrew's a postdoc. The PIs on the Minnesota side of this project are myself, Craig Sheaffer, and Yaniv Brandvain. And then we work very closely with the Land Institute, particularly David Van Tassel, who started the domestication work in Silphium, along with other partners at the USDA in Fargo, University of Wisconsin, Kansas State, Colorado State, as well as some international collaborators as well. So we have a big group that works on this, relatively big. So I will turn it over to Sienna.
Okay, sorry, I didn't mean to cut you off. Thought there was
that's good, Sienna. You better learn how to do that, you know.
All right.
Your PhD advisors can ramble on.
Okay, so I'm Sienna, and I will be talking about my research on the agronomics of Silflower. And I'm starting with this spring mowing study, using this as a management strategy. So this is about what Silflower looks like in the spring, and we are experimenting with mowing to see what happens when the plant regrows. So this is just a little diagram of kind of what it looks like. So we cut, oh man, there we go. In the spring, the control grows back pretty tall, usually around 2 meters, but there's plenty of variation around that. And the mowing treatments grow back a lot shorter, generally with more stems and a kind of more even flower height. And then there's also a difference in synchrony is the word that we're using, and that's just talking about how the flowers mature, whether it's at the same time or whether it's spaced out in time. So so that's the idea of, and the methods of what I'm doing. And then this is a picture of what the field actually looks like. So this is a randomized complete block design. So you can see it is random where the different plots are, but I'll show you which treatments we're looking at here. So this photo was taken the day of the last cutting treatment. So this is when all of the treatments have been cut, but they haven't started to flower yet, but you can start to see them growing back. So in the back here is that this final, the 4th cutting treatment on June 20th. Then in front, starting to grow back is the 3rd treatment cut on June 13th. And then growing back more the June 6th, and then in back was that 1st cutting treatment. So that's broadly kind of the premise of what I'm doing here. And then I'll show some results from this experiment. So it was planted in 2018. We took data in 2019 and 2020. Still working on processing the seeds and getting the data from 2020, but I'll be talking about 2019 data. And so starting just with plant height, we did see differences in plant height. So each box and whisker plot here is a different cutting date. And then this first one on the left is the unmown control plots. And then we did this in 2 locations in St. Paul and Rosemount, and also used 2 different genotypes. So there's a short genotype and a tall genotype. And there were a few differences like in height here, but those genotypes show a big difference in yield. So I'll show that next, that yield data. So we did see a pretty clear decline in yield compared to especially the later cut varieties compared to the unmown or the uncut varieties. But this first cutting date, the May 30th cutting date, wasn't significantly different. So it at least shows some promise as being a shorter, like more harvestable plant, more manageable plant, with a similar yield to the uncut plants. And so this is a promising finding, seeing that those yields are similar, and it's just good to know that we don't need to think about cutting later in the season. So thinking about cutting in June is probably not going to be that helpful. So part of why we're doing this is exploring whether that spring mowing could be used as a forage, and we're still working on that analysis of forage quality, but we're hoping that that all of these pieces could tie together and make this a viable planting strategy, or a viable management strategy. And okay, so I'm going to transition now to the variety trials that we have with Silflower. I have one experiment that I'll talk about, and then John Hill will also talk about a different variety trial. So this is a lot here, but I will walk you all through it. This variety trial was planted in 2018, and I use the term variety trial kind of broadly. We're looking at a bunch of different populations here and assessing them for yield. So we're calling it a variety trial. I'll start on the left. Well, first, so we're looking at yield in kilograms per hectare at these different varieties in 2 locations, in Isanti and St. Paul. And so on the left here, this bulk variety is what we're calling it. And this came from the Land Institute's breeding program that Kevin mentioned earlier. It was used in some previous agronomic experiments. So it's selected germplasm, but then it got bulked back together so that we could do some agronomic experiments on it. And that kind of bulk population is included in this variety trial. And then next here in the purple are 2 different wild populations from Kansas. Next up are what I'm calling Minnesota 1 and Minnesota 2. These are progeny from this bulk population. I should say these are selections from that bulk population, and they were identified as being relatively short and early maturing. And so John Hill made those selections. We collected the seed from those 2 individuals, so they're half sibling families. And then the last group of varieties are in green here, and those include selections from the domestication panel. So the domestication panel was a previous breeding experiment that was in Minnesota and actually throughout the whole country, but we looked at data on that panel from the Minnesota locations, and John Hill selected these 5 individuals that looked promising. And so that's what we were comparing here. I think a couple notable things from this data are just looking at the differences in yield between Isanti and St. Paul. So Isanti is in the Anoka Sand Plains region. It has very sandy soils with lower fertility than in St. Paul. So it's interesting to see that these yields are higher there. And I'm very curious to see whether that trend continues in 2020. And then the other notable thing here is I've pointed out this Minnesota 4 variety, which is also known as Crookston 202 or CR 202, sorry. And that yielded very high or relatively high in St. Paul and in Isanti. So those are 2 exciting pieces of this data that I'm curious to see what happens next year too. But this is kind of the start on our variety trials, and then I will pass it over to John Hill, and so that he can talk about a different variety trial.
Was the population on the sand plain irrigated?
Not very much. So we used irrigation with planting, and making sure that the plants got established. But thinking back, I don't think there was any point in the growing season that it was irrigated in year 2 and year 3.
Well, that is very exciting results, because a number of us have been thinking about Silflower as a new oilseed crop for the sand plain. So that's a really interesting initial results.
Yeah, thank you. So I think we'll keep going with the presentations, and then we're hoping to have some time at the end for discussion. But please feel free to jump in with questions in the chat or as we present too.
Okay, so hi, I'm John Hill Price. I'm a grad student in Kevin Smith's lab. I work mostly on silphium breeding and genetics. So I'm going to talk about our second generation variety trial. So this was planted also in 2 locations, but in Rosemont and Crookston. And it contained 13 half-sib families. So that's kind of the unit on which we're doing a lot of our trials at this point in time. 3 of them, these CR 202, 252, and 292 were also in the yield trial that Sienna talked about, so we have some overlap there. And then these 3 blue ones, the RM ones, were some of our more advanced selections for reduced height and early flowering. They're descended from that kind of same population Sienna talked about, but they're more generations advanced. These teal ones on the left, the B ones, are kind of parents of those blue RM ones. So we planted them in the same field to see what kind of gains we've made for selection. And then the orange ones on the right are advanced selections from the Land Institute. So David Van Tassel sent a few of his best varieties for us to try out. We're still processing yield data on these, but I just wanted to show some data. So this is height that we measured. We measure height, we measure maturity, we measure a few other traits of interest to see where they are. And one encouraging thing is the blue ones, the RM ones that we've selected for reduced height, did in fact have reduced height. So that's something, and stay tuned for more yield data. Next slide. So then I want to talk a little bit about just some of the kind of more generally the breeding work that we are doing, what our process is, just for some context. So a lot of our selection and evaluation and crossing work right now is happening in our crossing nursery, which is located in Rosemont. This is a picture of that field taken before flowering in 2020. So this field was planted in 2019. It contains about 40 different families. Our 2020 iteration contains about 25 more families, and we'll continue adding as we evaluate more stuff. Next slide. So then in this population, we're looking at a whole host of different traits. These things include height. We've harped on that a lot. Silflower can get pretty tall. So we think reduced height is probably an important breeding goal. We look at date of flowering, we look at stalk diameter. We have some data to suggest that that is associated with yield and also with harvestability. We look at things like flower diameter, number of seeds per head, that's highly variable. We can also use those to predict what the weight of a seed will be for an individual plant. We also look at things like maturity and shattering. So these pictures show on the left just variation in flower head size. This is from Kansas, and then the right just shows kind of variation for shattering on a single plant. But silphium doesn't have an explosive shattering response like wheat or barley, but it will eventually lose seeds over time. So it's something we're paying attention to. Next slide. So then some of our next steps, the things we're thinking about moving forward. One, we're looking at trying to figure out the best way to evaluate crosses in our variety trials or yield trials. So we make crosses in our crossing block, and we're trying to figure out what to do with them. But moving forward to yield trials seems to be the logical next step. We're also looking at developing larger populations. So we might combine, if we look at a variety trial and have 3 or 4 good varieties, we might mix that seed together and try it out as a variety. We're still once again thinking about how to do that well. Looking at increasing the size of our yield trials, both in terms of number of entries or number of replications per entry, / number of locations. Both of them have only taken place in 2 locations so far. So we're trying to figure out the best way to expand that. And then finally, we want to get oil quality and percentage analysis. We're currently working with the USDA and Fargo to make that happen. So we don't have that data yet, but hopefully we'll have it in the near-ish future. Next slide. Okay. So then my next talk is about measuring inbreeding depression in Silflower. So this is a summary of a paper that's currently under review and hopefully we'll have out pretty soon. But next slide. So inbreeding depression is the kind of general tendency for offspring with parents that are related to be less fit or less healthy than offspring with unrelated parents. And we expect to see this in Silflower because, as Kevin mentioned, it is an outcrossing species. In nature, it doesn't self and it tries to avoid mating with its relatives. When you do breeding, you're almost always going to have some level of inbreeding. When you cross, good things by good things, they tend to be related. So we really need to get a handle on what kind of impacts this might have going forward. So in this study, we measured 2 different flavors or kinds of inbreeding depression. The first one is seed mortalities. So when you mate related individuals, some number of those offspring will not survive to maturity. In our case, they don't even make it to a fully formed seed. So the kernel or the embryo dies soon after fertilization and you have just an empty husk. So you can look at that as a fitness hit. It's also kind of a yield hit. So in our studies in Silflower, we found that if you mate full siblings, just due to seed deaths, you see about a 20% reduction in seed set relative to an outcrossed individual. So that's not including self-incompatibility. We dealt with that. So this is just due to inbreeding depression killing some number of seeds. So that's a lot. That number also, we can also compare that number to other similar wild species, and we find that Silphium is relatively low compared to some other perennial outcrossing prairie plants, but still kind of on the higher end for a lot of crops. One kind of practical way we can think about this is we can imagine if you took a bunch of seed that was all from one full sibling family and planted out in an isolated field, it could only mate with itself. As I said, you would imagine that you would have a 20% reduction in seed set or yield due to just seeds dying. And if you have a family of half siblings, they're half as related as full siblings, that effect would be cut in half. So this graph on the right kind of summarizes what you could imagine. This is all simulated data, but if you planted out that some number of imaginary families, what the yield hit would be due to inbreeding depression. So basically, every family you add, you cut the penalty in half. What that tells us is by the time you get to 3 half-sibling families or 5 full-sibling families, your yield hit is only about 5% of what it would be as an outbred. So that's something we can use in breeding to say when we release a variety, we need to make sure it has a certain amount of diversity, and this tells us what that is. Next slide. So then the other way we can look at inbreeding depression, and this is maybe the more classical way that you might be familiar with, is to look at effects on traits. So a plant survives, makes it to maturity, but it has some sort of reduction in number of flowers or number of seeds it produces or height or something like that. So we also measured that. There's a lot going on in this figure, but what it basically shows is each line is a trait. You can see these here on the left-hand side. Each dot represents a family or a family's value for that trait. And then the units are basically percentage of inbreeding depression. So if you're at 0.75, then you are 75% worse for that trait than you would be as an outbred. And then the circles are highly inbred individuals, and then the triangles and squares are less inbred. But what this shows is we have a huge reduction in yield. So this is different than the seed set decrease I talked about earlier. This is due to plants not producing as many flowers usually. You can see you have about a 75% reduction in flowers for the highly inbred individuals, less severe for the less inbred individuals. And then we also see severe reductions for things like height, number of stalks, and stalk diameter. So this is, once again, information that we can use in breeding. This is not to say that we can't domesticate Silphium. There are plenty of crops which have inbreeding depression problems, but this means that we need to be clever about the amount of diversity that we have in our populations and how we manage them moving forward to make sure that we're not massively hurting important traits through inbreeding depression. Next slide. Okay, so then my last little talk here is about efforts to build the first linkage map for Silphium and some work that we've done with self-incompatibility. So Andy, who's going to talk later, also was a major contributor to this work. Next slide. So one practical outcome is we built the first SNP-based linkage map for Silphium. The mapping population was about 280 individuals. We have about 200 markers that settled into 7 linkage groups. Silphium has 7 chromosomes, so we'd expect that. We've had versions of this map around for a while. So if you've been at previous presentations, you might have seen a version of this. This is the final updated version that we're working on the publication for now. So this is something that is good for its own sake. It's a nice first step in building a genome, in marker-assisted selection, things like that. We also were able to one practical thing we were able to do with it was to do QTL mapping. So we found significant QTL for stem color here on chromosome 1, for leaf thickness on chromosome 5, and then speed of germination, so how fast a seed, the root emerges on a seed, on chromosome 7. All of those might get used in breeding. The speed of germination one especially seems to be pretty high effect, but it's more useful just kind of as a first step, or oh, this is a thing we can do in Silphium. It opens the doors for future work to do marker-assisted selection. But one other thing that's let us do that I'm particularly excited about, and I'm going to talk about for the rest of this presentation, is that we are able to map the self-incompatibility locus in Silphium. So that's the blue one on chromosome 6. Next slide. So a little refresher or new information. Self-incompatibility is a system whereby plants can keep from mating with themselves. So if a plant has a pollen grain laying on its flower that's from either itself or from a closely related individual, it has a series of mechanisms that says, oh, this pollen is too related to me, don't let it fertilize the seed. Using these systems, plants avoid inbreeding depression, like we talked about in the previous presentation. So Silphium is self-incompatible. You can't self-pollinate it and get seeds, or you can get a few, but not many. And in Silphium and in other species which have what's called a sporophytic self-incompatibility system, this is controlled by a single part of the genome called the S locus that contains 2, or probably more than 2, but at least 2 genes, one of which is expressed in the female, one of which is expressed in the male. And when those 2 meet, if they are from the same allele, they have the same genotype, basically, that triggers the self-rejection. And if they're different, then the thing can be pollinated. So you can kind of think of it as like a lock and key system. So this is a really interesting system just from a biology standpoint. A lot of people like studying it. It's really cool. But actually finding where in the genome this S locus is a fairly tricky problem. And it's something that has, it has been done in a number, in a set of other species. I can't remember how many, but not a ton. So any more that we can do is good information. And then also has potential breeding applications. If we can look at the genomes of 2 plants and predict whether or not they can mate, that could be a useful thing moving forward. Next slide. So very briefly, we developed a new set of experimental design and analyses methods to map the S locus in Silphium. So kind of very briefly how this worked is we had about 280 individuals that we made our mapping population from. We took a subset of about 84 of those, and we wanted to try to find out which S allele all of these 84 plants had. And a way you could imagine doing this is you could intermate all of these 84 plants in every possible combination. And then you could say, well, plant 1 and plant 2 were able to mate, plant 1 and plant 3 couldn't mate, so on and so forth. And based on the pattern of mating and not mating, you could say, well, these things are the same, these things are different. However, if you want to mate 84 plants in all possible combinations, that's about 7,000 crosses, and we didn't have time for that. So what we did instead was we took a very small subset, about 260 crosses, which is about 4%, I think, of those crosses, and actually made those. So this figure kind of visualizes that every tick is a plant. That means every square is a possible pairing. And then the ones that are either red or blue are ones we actually made. The red ones were incompatible and the blue ones were compatible. It was basically structured in these little groups, and then we linked them together through single crosses. And then Andy and I worked together to develop a couple of different statistical methods to use that little bit of information to infer what the rest of the things are and what the groupings are. And it worked. So this is only the second time in the Asteraceae family that an S. locus has been successfully mapped. Our method also requires a lot less resources and a lot less time than previous methods. So we think that it's going to open up research into this phenomenon in a lot of other species. So we're really excited about it. And we're excited about this is a model of what forever green can do in kind of bringing together a biological question and an applied breeding question to like make something new and exciting. So thank you.
So it looks like we've got Yuki next. Yuki, do you want to get started?
Yeah, sure. Okay, so good afternoon, everyone. So I'm working on the micropropagation side of this whole thing and trying to reduce this Silphium's juvenile stage. So if we can go to the next slide. So in any kind of breeding program, one of the main factors that drives the breeding cycle time, like the time it takes to make a new generation, that is the juvenile phase of the plant that you're working with. So that's like that's basically the stage between planting the seed and getting it to flower. Like that amount of time determines how quickly you can create new populations. So if we reduce this juvenile phase, then we should theoretically be able to achieve a faster breeding cycle and get more genetic gain per unit of time that we have. So Silphium is a perennial, and they flower in around their second year of growth. So they first grow for 1 year where they just form a rosette. They're very close to the ground, and they don't produce any stems at all in the first season. It's just like a big plant on the ground. And then in the second year, they will actually bolt in the summer, and that's when they will flower. And the way to reduce this juvenile phase, there could be several ways, but one of these could be vernalization and gibberellins. So vernalization is the process that Silphium goes through, in which they have to experience a cold period. After the end of the cold period, that's when they can flower, and this is the winter. This is the winter that they experience in nature. So vernalization does not necessarily need to be the entire length of the winter. It just needs to be long enough, and then the plant just kind of is dormant and just kind of waits it out until the winter is over. So vernalization shouldn't be as long as the actual winter, and I'm trying to maybe find a specific length that would work. And another one is gibberellins, which is a plant hormone, which can also reduce the plant's juvenile phase. Another way of helping with the overall breeding program is micropropagation, which is growing a plant in a test tube. Micropropagation is very efficient and cost-effective and allows us to produce a bunch of disease-free clonally propagated plants. Now, being able to produce clones of a certain genotype is very important for us because Silphium is an outcrosser. So getting self-seed is very, very difficult, and getting genetically identical seed in that way is might not be possible. And the current ways of clonally propagating Silphium, such as division and cuttings, are both very slow, and especially with division, you have to sacrifice the mother plant by dividing its crown, and the resulting plantlets will take a while to kind of recover from that damage, that shock, and be able to grow. So being able to micropropagate it will be very efficient for us, because that allows us to produce much more plants at a much faster rate. It also opens up new avenues of research in vitro, such as mutagenesis or embryo rescue. And we can go on to the next slide. So I'll talk a little bit about my micropropagation protocol here. So we do know that genotype, for most species at least, the genotype has an effect on the way that the plant responds to in vitro conditions. So as such, I'll be selecting 5 different genotypes from the UMN domestication panel in Rosemount, and I'll be using crown tissue and stem cuttings as the tissue for micropropagation. And I also want to determine the best concentration of cytokinins, which are plant hormones that induce plants to make more shoots, and auxins, which are plant hormones that induce root growth. And I'll try to determine the best concentration of both of these hormones for silphium. Now, because silphium has never been micropropagated before, and there's not really any, well, there are related plants to silphium that have micropropagated, such as sunflower and chrysanthemums, but that relation is pretty, they're not that related, so is what I'm saying. So I'm doing this from scratch, so I'm using a bunch of different cytokinins and auxins. I'll be doing 3 different cytokinins, which are BAP, benzoaminopirin, zeatin, and kinetin, and I'll also be doing a whole bunch of auxins, because there exists much more auxins than cytokinins. And due to the virus, as well as problems with getting the material, and also just cleaning the material, because I need these plant tissues to be 100% clean for this to work. Due to that, I've not yet started on the experiment, but I will be starting on it in the coming weeks. Can we go to the next slide, please? Thank you. The next one is that I would like to determine an exact way of vernalization, well, like an exact length of vernalization that will be good for allowing silphium to flower. I will be using seeds from the UMN crossing block, also in Rosemount, and I'll be also using 5 genetically distinct genotypes. And I will grow these in the greenhouse and produce 6 different age groups that are based on the number of leaves. So I'm not using the chronological, like the age, because this little picture on the right here, as you can see, all of these silphium, these were started from seed. Even though they're all started from seed, they're the same age, but as you can see, some of these have, they are much more developed. Like this one has a very large leaf, and these other ones on the right here still have their cotyledons. So that's why I'll be using the number of leaves to get a kind of a uniform physiological development. And then I will grow these at 4 degrees, I'll vernalize these at 4 degrees Celsius at different lengths, with 0 being the control, and then the rest from 1 through 6 weeks. And then I'll grow these in the greenhouse and see whether they are able to flower. And yeah, so that is, I just started this experiment. I have the seeds. I have my seeds in the cold chamber right now, because the seeds need to be stratified before they can be planted. So I'm beginning this one as we speak. And yeah, that is all I have. Anyone have a question? I can answer that. Well, actually, there was another part of the experiment in which I'll be looking at how gibberellins affect the silphium, but I thought that was too over time, so I didn't include that. But basically, for that experiment, I'm applying gibberellins onto the silphium plants at their meristem, because that's where they respond to gibberellins. I'll be applying gibberellins to see whether that can reduce their juvenility phase as well. So that's the other part I just didn't include. But yeah, if anyone has any questions, just feel free to ask.
I think I'll go ahead and move us on, and then we can keep thinking about questions at the end. So,
all right, thank you.
Okay, so I'm Andy Rudusky. I'm a postdoc with Univ Brandvein working on this. So in addition to all the agronomic and breeding stuff that you guys have heard about so far, we've been making a lot of progress towards characterizing variation in natural populations too. Advance the next slide, please. So we think this is important for a few reasons. First of all of the breeding improvements and phenotypic responses that you guys have seen so far, these have all been done in experiments using germplasm that's derived from collections from a very, very narrow geographic range. So if we're trying to de novo domesticate a wild species in its native range, it's useful to know how much variation is out there, and then also like what's the relationship between geography and genetic variation too. So the next few slides, this is just a brief summary of work that we've done characterizing population genetic structure within the species. And this has been written up and accepted to the American Journal of Botany, and it should be in press very soon. So silphium's genome is very large and repetitive. So it's difficult to assemble, and that makes it really difficult to get consistent, reliable genetic markers for many different individuals from across the range to look at the types of things that we're interested in. So we made a high quality transcriptome from a bunch of different tissues from a single individual from the Land Institute's breeding program, and then used this as a genotyping reference. And we resequenced the transcriptomes of many plants from across the species range. So those resequenced plants were collected from seed and then grown in a common garden in Kansas. Next slide, please. So the big take home picture is that it looks like Silphium is made up of 3 genetic clusters across the range. So on the left, these are our sampling locations in colored shapes. And then the gray shading underneath the shapes on the map is our estimate of habitat suitability. So this, you can think of this as some kind of estimate of the species range. And on the right are the results of a genetic PCA. And so this, it forms 3 tight clusters. So we define these clusters based on how they correspond to geography. So the squares in the upper left of the PCA space, those are plants from Kansas and Nebraska. The bottom of the PCA space, the triangles, are plants from Texas and Oklahoma. And then plants from Mississippi, Arkansas, Missouri, Illinois, and Wisconsin all cluster in the top right of the PCA space, and we call these the eastern region. So there are no clear physical boundaries between, like geographic boundaries between these guys. And so if you look at a plant from Missouri, it's geographically very close to Kansas and Oklahoma populations, but genetically it doesn't cluster at all with those at all. And it clusters much more tighter with plants from like Illinois or Wisconsin. And it's also worth noting that going into this, Silphium is this iconic prairie species. And so we've kind of assumed that it originated within the prairie. And yeah, so going into this, we thought that the center of diversity was going to be Kansas or so. Okay, the next slide. So we do a little bit more analyses. We look across samples and we say, okay, how many different ancestry layers are there in all these samples? We get 3. And then the relative proportion of each of these ancestry layers within each individual is kind of defining too of its geography. So in the top right-hand part of this slide, this kind of bar chart, sideways bar chart, plants from the east overwhelmingly have this 1 layer of ancestry that's denoted in yellow. Plants from the south have a different layer of ancestry that's dominant, and that's green, but they also have a little bit of this orange ancestry. And then plants in the west have this different, this inverse of the south, basically, where they have a lot of orange ancestry and a little bit of green. We can build phylogenies of these samples too, and it clusters in the same way. If we look at the nucleotide diversity within each region, we find that the east and south have comparable amounts of diversity, but the west has really reduced diversity. And that's interesting because all of the breeding program germplasm is collections out of the West. And so it seems like all of the improvements and advancements that we've seen in the breeding program come from places where they have reduced genetic diversity. And also, if we look at just the amounts of diversity within populations, so breaking down even further, not just regions, but within populations, we see that the most diverse populations are in Mississippi and Texas. So taken together, all of these results suggest that the geographic origin of Silphium, it was kind of in the American Southeast, so Mississippi. And we think that there was some kind of dispersal event early on into Texas and Oklahoma. And then those Mississippi populations kind of expanded its range north. And then there's a secondary bottleneck from Oklahoma into Kansas and Nebraska. So this is really unexpected. Like I said, we thought that Kansas would be the center of diversity, but that's not what we found at all. Next slide, please. And then these results have really changed the way we've thought about collecting. So just recently, over the summer, some folks from the Land Institute went out on another collection trip, and they focused exclusively on the American Southeast because of our results here, and because these are super diverse populations. And so they've done a lot of collections there. And so we're really excited to see what type of variation we see in these new collections. And that's an ongoing. Yeah, that's all I have for this. And I think Kelsey's next.
Hi, my name is Kelsey Peterson, and I'm working on doing a PhD in the Brand Vein Lab. And I very luckily have this natural segue from what Andy was talking about into some applied uses of how we're beginning to look at these wild populations. And Andy described several distinct populations, and I just want to note that this is the very beginning of understanding how that diversity might be useful in a breeding program. But we're going to switch gears just a little bit. And I'd like to mention that every major crop that we're using in the world for food or forage or material has some kind of pest and has some kind of pathogen. Silflower is no exception. And as Kevin spoke to us earlier about where Silphium is and where we're seeing pests and pathogens, we are seeing a lot in Salina, Kansas, which is where the Land Institute operates, and not that many in Minnesota. And we don't know how long that's going to last. So one of the goals with the breeding program and with domestication of the crop, which I imagine is going on simultaneously, thanks in large part to the University of Minnesota, we acknowledge the need to find disease resistance and find strategies to mitigate pest damage. So along with other strategies being developed, including the use of fungicide and pesticides and timed mowing, actually, thank you, Sienna, for your work. I'm investigating the utility of wild populations for traits that could be included early on in the domestication process that could provide useful resistance. This could overcome barriers that we're facing now with with Silphium, and also get it to farmers faster and potentially introduce this resistance really early on. We know a lot of modern crops are using wild relatives or secondary and tertiary gene pools to search for this resistance. They're kind of going back in time, especially to confront diseases that are becoming more and more problematic in fields. This takes a lot of time and a lot of energy, and it's not very easy depending on the genetics of the organism and outcrossing and incorporating traits into inbred lines. So there's a big advantage, we think, in doing this early on in the domestication and breeding process for Silflower. It's kind of this preemptive front loading of resistance. Finally, this work might also help us maintain variation for traits that we can continue to pull on as Silflower progresses into more fields and likely into more challenges. So how do we look at this variation in these wild populations? Sienna, can you click? Thank you. So I'm going to give a really brief overview of how we're looking at 12 different populations from these regions that we're calling the West, the Central, and the East. And these locations are along a rainfall gradient, which has a lot of ecological importance and is also in this area that we might see Silphium soon in either breeding programs or in farmers' fields. So just a brief explanation of reciprocal transplant. We collected 12 distinct wild Silphium populations and planted them in 3 distinct locations within the regions where we did the collections. So you can see the diagram kind of explains the reciprocal transplant design, and the circle, the triangle, and the square are where those plantings took place in 2019. So some other exciting things about this reciprocal transplant study is, Sienna, can you click? It's in major collaboration with 3 universities. University of Kansas pulled a huge amount of effort at the front load of the project. University of Minnesota, the Land Institute, and there are plots planted at the University of Illinois campus. These are pictures of this reciprocal transplant design, a satellite imagery and an up-close field image. And so why are we using reciprocal transplants? Studying wild populations is hard. If you were to walk into a prairie and look at wild plants and one wild plant is doing great next to another, it would be really hard to discern why that plant is doing great. Is it because of the genetics or something about the environment? Maybe the soil is just better where that plant that looks good is. So when we remove these plants from the wild and we put them in these different locations, we can track traits of interest such as height or number of trichomes or disease susceptibility in all 3 locations to really narrow down what about that plant is allowing it to show this trait. Is it the environment or is it its genetics? Sienna, can you click? So these are just some preliminary images to show you what we are seeing in the field. These pictures were taken this year. The two smaller pictures of the clearly disease susceptible and the disease resistant plant were taken previously. But so these stands of Silphium, which are labeled Western and Eastern, come from those extremes of the reciprocal transplant design planted in the central location. And you can see that these plants are, it's easy to see that they're planted in the same field, but what you can't see is that they're planted right next to each other and they look really different. I couldn't tell you why yet, but with several years of data, we might be able to kind of get into why the Western plants always look spindly. Maybe they look spindly in all the locations, or maybe the Western plants just look spindly in the central, or vice versa with the Eastern plants. In addition, back to this disease susceptibility and resistance question, we are seeing that there is a difference in the source prairie location to these different planting sites in the plant's ability to handle various diseases and pests that we know Silphium is going to encounter as we move the plants from their wild populations into breeding situations. Okay, you can click. This final slide is kind of this extra layer of information that we're gleaning from this reciprocal transplant design. And what we're looking at here is if you take the same plants and you plant them in different locations, that plant material might look really different in one location to the next. And how we measure that is going to be really important and could save us time as we're moving. Let's say like the Western Silphium plants, that is mostly what's grown in domestication panels across the Midwest. As we've already seen, Silphium grown in Salina grows differently in Minnesota, perhaps better in Minnesota. And those are trends we'd like to be able to measure and track easier and better later on. So those are the multiple layers of this really large project that I just skimmed through. And I'd be interested to hear if anybody has any comments or questions about it at the end of all the presentations. Here's a natural segue into yet another layer of this project where we're looking at R genes or resistance genes, and Andy is going to talk to us about that. You can click. Thank you.
All right, yeah, so these last two topics are kind of updates on resources that we're building that will either complement ongoing projects like this resistance gene stuff or provide new opportunities too. So kind of piggybacking on what Kelsey said, if we assume that across the species range, different populations experience different pressures from their pests and pathogens, we hypothesize that if we look at variation in the genes that plants use to respond to those different pressures, that looking at that variation in those genes across the species range will be helpful and informative too. Can you advance the slides, please? Thanks. Okay, so the genes that we're interested in are called these resistance genes or R genes, and they belong to the family of these NLR genes. And they have this very specific type of structure, so you can identify them based on these domains that they have. And we're interested in sequencing these from across the species range. So the technique we're going to use is just a basic hybridization capture technique where you build baits for a target. So the target in our case would be these R genes. And you build baits that hybridize to those targets. Then you apply beads to the baits. Those beads are magnetic, and so you can pull down sequences that have the baits and the beads in your sequencing libraries. And the end result is you have a sequencing library that's enriched for your target sequence. So in this case, it would be these R genes. However, we don't have a very good genome assembly yet. And so that is a problem for us. However, because the R gene structure is conserved within this family, we're using that as an advantage to us. So the next slide, please. So this technique is often done in model systems where you have a genome and say you have a handful of annotated R genes. So you can build baits based off those annotated R genes. And in model systems, usually you have, this is often done by sequencing different lines of the same species, and those lines might be susceptible or resistant to some very specific disease pressure. Then you can then sequence and map or clone the gene that's responsible for resistance for that very specific disease. One good thing about this is because these genes are conserved, let's say a model organism has a handful of annotated R genes, you build baits off that, the data that you get back from the sequencing is often greater than the annotated R genes that you started with, because of the conserved nature of these genes too. So you end up pulling down more genes, more different types of genes within the same family than you started with. So you can, the next slide, please. So although we don't have a genome, we do have some sequence resources. And so we've gone through the transcriptome from Silphium, some sequencing that we have done, as well as related species such as sunflower and lettuce in the same family that have good genomic resources. And we've been able to identify a lot of sequences that look like these R genes. And then, so we're in the process now of building baits based on the sequences here. And then we're gonna do this RenSeq protocol on 100 samples from a dozen populations, which are the same populations that Kelsey just talked about here. So we're looking at, interested in looking at the variation in these genes in wild populations. All right, and then the next. Okay, and then finally, we've talked a lot about the lack of genomic resources in this. And so this is just our plan. We've secured funding from the Land Institute to put together a genome. And so this is just a brief overview of what our plan is for that. Next slide, please. Yeah, so far, John Hill's linkage map has been really helpful, and we've made a lot of progress using that and some transcriptome markers. But a genome assembly will really allow us to have more fine-tuned control over marker-assisted selection. We can do GWAS, we can do finer mapping of traits, and it'll allow us to really have a consistent set of markers that we can use in a wide variety of circumstances. So next slide. So the general overview of what we're going to do, we're going to be using what's called a trio binning approach. And so in this approach, we'll be sequencing 3 different plants. One of those is an interspecific F1 hybrid. The 2 parents of that, one will be Silphium integrifolium, which is the species of Silphium that we've been talking about all day today. And then the other parent would be its congener, Silphium perfoliatum, which also has a lot of breeding work being done on it as well. And so the way this approach works is that we'll long read sequence the F1 hybrid, as well as short read sequence each parent. Then if we look within the short read data, we can identify sequences that are uniquely specific to either parent. Then we can look in the long read data of the hybrid for those uniquely parental sequences, and that'll allow us to separate the long read data into 2 different sets, one belonging to each parent, and then we can assemble those long read data into contigs for each individual species in the parental species. Then we're going to do some high-C sequencing to further arrange those contigs into larger chunks, and then do some transcriptome sequencing of each parent as well to annotate the genome. But the end result of this is that we end up with 2 genomes for the price and work of 1 assembly. So we're really excited about this, and we're expected to get this started relatively soon, within the next few weeks. And that's all I have.
All right. Well, so you heard a lot of different highlights across a wide range of work that we're doing with Silphium, but now we can open it up for questions that people have for any of the speakers.
Would it be helpful if I keep the slides open, or should I stop screen sharing?
I don't know.
Okay, I'll leave it open for a bit.
Keep it up and see. We'll see.
I'd like to congratulate you guys. You guys have really put together some great research, a great project, and a great collaboration across a number of different areas. It's really, really impressive what you guys have been able to do. One question that I have is going back to some of the breeding work in terms of the barriers to increasing yield. what is the idiotype of the flower that you're looking for in terms of, the seeds associated with the ray flowers rather than the disc flowers in this species? So what could you talk about the limitations of seed set on a flower head or on a head? Do you have to increase the diameter of the head to get increased number of ray flowers? What is the general thought about that?
Well, so I mean, I'll let John Hill can speak about that because I know he's thought about it a lot. But yeah, you're right. So because of the way the seeds are arranged, there's a limit to the number of seeds that can be produced on a flower. So if you want to increase yields, you're either going to have to increase the number of flowers, or you're going to have to increase the size of the flower, to get larger or more seeds. So those are the sort of two potentially different idiotypes, I guess. But John Hill, do you want to add to that?
Yeah, so we have, it's a good question and one we're thinking about a lot. And like Kevin said, we kind of think about yield as being the composite of number of flowers, number of seeds per flower, and then weight of seeds on an individual flower. So you multiply all those together, right, and you get yield, right? And one kind of tricky thing that so the number of seeds per head or number of ray flowers per head is highly variable. We see anywhere from less than 20 to over 120. And we have both of those within our breeding program. The problem that we see is that as you increase the number of seeds per head, you'd feel like you would increase yield, but what you often end up doing is you actually decrease the size of individual seeds. So it's got 120 seeds, but they're all very thin and small and mostly husk. So we're interested in, we're focusing on one, whole plant yield and kind of just like, let's see what different routes the plants take if we select for this trait. But then we're also selecting for flower diameter, like Kevin was talking about. So if we get bigger flowers, we can also then get more seeds per head, but then those seeds can be bigger. And then we're also selecting for weight per seed. So we're kind of selecting for all these traits independently, and then we'll either recombine them or just kind of see which route wins.
Are the genes that control head size, if in annuus, right, there's a huge difference in head size, right? Are those genes known that control head size that could then increase the area for the development of ray flowers? Are those genes known?
In sunflower or silphium?
Well, I would guess there may be some relationship, right? Are they known in sunflower?
Andy, do you know? Or
I don't know off the top of my head. I imagine that.
I don't think genes. There might be like QTL, but I don't know that they've been mapped or refined down to the gene.
I see. All right.
Yeah, and we're still there's also the broader question where we are definitely still figuring out how useful sunflower information is in silphium. There's some ways in which we're finding it to be very interesting and useful, and then there's other ways from which it's maybe less so. So we don't. Yeah, we're still trying to work that out.
Yeah. Well, I have one more question, but I don't want to hog the questions. But what is the disease incidence in the area of diversity in the southeast? Is there, and within that population, is that the site where you're identifying disease resistance?
So we don't have a good understanding of the disease situation in the southeast. The populations that we're looking at for the resistance genes are not from down there at all.
That's what I mean. But based on what you guys have learned, that would be a place to start looking, right?
Yes, yeah. We kind of anticipate that there's gonna be all sorts of variation down there that's gonna be useful.
Yeah, right.
This is all very, very new.
Well, for me, you know, when we first started putting this program together, you know, there was this big debate whether the basic genomics and population work should be funded. But I think it's now a damn good thing that the argument made was for this type of comprehensive approach. So I think it's really, really exciting that that the group made that agreement very early on.
Yeah, I agree.
And I'll say that at least we're working on getting wild stuff into our breeding program right now. We've got wild plants in the greenhouse that we're crossing with kind of more advanced stuff. Yeah. So it's undergoing, or it's happening right now.
Yeah. Cool.
I have a question for John Hill and Andy. First of all, what a holistic breeding program. So expressive and nice presentation. Thank you for the presentation. So my first question to John Hill is about we have huge amount of option as to the crosses, and we test a smaller sample of it. And Kevin already published paper with our package called Popular. They predict which population will produce the most superior progeny. It was very good. When I was a soybean breeder, I used that. But the other breeders always wonder, how will you be able to validate or confirm the population you made is works and the one you haven't make now works? So that's first question. What is the plan you will validate the popular, or will you develop a different popular version for different crop? Or for each crop, how will you validate the populations you never made? They won't produce nice progenies.
Or Kevin, do you? I have some.
Go ahead.
So that's a good question, and we've not really interacted much with, or we have not used PopVar at all in Silphium. And kind of the funny thing about Silphium is because it's so new and we have so much diversity that there's almost, how can I say this? There's a way in which it almost doesn't matter. In that it's, you know, if I cross any two individuals, I can guarantee that they're going to be fairly different, and the family that I produce will have a lot of variation in it. So there's ways in which, you know, something like barley, you have to work really hard to maximize the amount of variation that you're getting. But something like Silphium, especially because it's outcrossing, everything's highly heterozygous. A lot of things are unrelated. We have so much variation for traits of interest, like I would imagine probably orders of magnitude more variation for a lot of our kind of key traits than something like barley or soybean, just because we've not gone through, you know, so many cycles of selection. That, yeah, we're not really in a place yet where we're worried about kind of maximizing variation. We just kind of trust that anything we cross is going to have a lot of variation. As we get down the road, as we develop more genomic resources, as genotyping becomes a routine thing in our program, I think we'll definitely be thinking about that, trying to maximize the variation of our crosses. But right now, it's just kind of hope.
So how will you choose that smaller percentage of sample to move forward? How did you pick that out? For example, I'm working for alfalfa breeding. I have a lot of potentials. What is the way you pick out like 1% of potentials to move forward?
Yeah, that's also a good question. We've not really had a very rigorous methodology. It's been kind of a very much just going by feel. We have kind of identified a subset of traits that we think are worth putting effort into now. So, you know, every year we maybe advance a small number. I don't have a number, but a small number of crosses that are kind of focused on that trait. Another thing that we'll do is we've made So for when we were selecting for reduced height and early flowering, we just made a bunch of crosses. We developed a large diversity population, put out in the field, and then I just walked through and picked the bottom, 5 or 10% for reduced height or yield. So kind of a low percentage like that, but just from a diverse population. So sometimes it is just what are the best things for the trait that we're interested in, making sure that they're coming from different families and diverse backgrounds. I don't know how well that answers your question though. I guess the short answer is we don't have a super solid methodology. We've kind of just seen what happened.
Thank you. Yeah.
I think another thing that might be worth considering is that, a lot of this, the selection is based on individual plant characteristics, right? So we're selecting on individual plants, which, we may be different in terms of its effectiveness compared to selecting on a plot basis. So ultimately, when we grow these things in an agricultural setting, they're going to be growing as part of some sort of community. It might be a monoculture of Silphium, or it might be a polyculture with other species mixed together. So one of the things we have to do is develop the ways that we phenotype things in order to make selections and make crosses. So the easy thing is to start with individual plants. We can grow hundreds of plants and then go find the short one. The harder thing is to figure out how you grow these things in the cropping system that approaches what might be used in agriculture, and then do selection on that to make crosses. And we're not there yet, but we're working on moving in that direction to get things into that sort of a setting.
Thank you, Kevin. That is a difficult question. I will ask a simpler one for Andy. So Andrew, you present the germplasm diversity. I see your PCA1 is only 9%, PCA2 is only 4%, but your separation is so wild. What is the magic over there?
What is the magic? Oh, I don't know what the magic is. Yeah. So 13% of the variation. It's not a lot, but when it's separated so strongly, it doesn't require much, I guess, when the separation is so strong.
Well, when I see the figure, you can see the Texas, Oklahoma. They are 100% can be separated with Nebraska, Kansas, etc. Yeah. So the PCA to me is needed to be over 60 or 70 to have a clear separation. And with 9% and 5%, you separate so well. Just wonder how you did those magic. Okay, so thank you.
All right, other questions? Well, if we don't have any, Don, at the beginning, I told him that usually the beginning of the meeting is when you show up and tell everybody some earth-shattering news from the Capitol or whatever. And I told him that I can't provide that kind of information because I'm
well, I sent out a note to you guys to send me that information and to report to the Capitol. So I won't mention it openly what it is, but return what I asked for as soon as possible. Thank you very much. Very impressive for, across the board. So thanks again for all your good work. And so next week is what? Thanksgiving. So we'll see you.